So let me give you an idea of what I'm hoping to cover in this 40 minutes or so. Get my watch going there. So kind of initially, like, why bother doing modeling? A little overview of the kind of the classical methods that we use. Look at them in particular for reversible tracers and irreversible. Talk about those, how we develop a tracer briefly, and then how we use those tracers, how we, for example, using modeling to separate blood flow or binding. How can simplify things? And where a lot of my work's been in the brain, the challenges of working outside the brain. And then a big part is really going to be looking at when do we need it, when might we be misled by some of the simplifications that we have. And kind of finally, I can't miss the opportunity with Terry and Ramsey involved here to talk about our next generation brain imager, what that might open up, and some maybe closing thoughts a little bit there. So first, maybe why model? And of course, just be sure we're all on the same page. We're talking here about cyclotron producing isotopes, handing it to our radiochemist to produce a radiopharmaceutical, where we'll inject in a subject, and usually for the purpose of a PET modeling study, collecting dynamic data over time, 4D data in ideally calibrated units, becquerels per ml. And PET is very unique because it is based on the classic radio tracer method, because we're using small quantities of radiopharmaceuticals. We're not altering the biology. We're fortunate to have isotopes that are easily labelable, and then any chemist should fairly forgive me for that one. The half-lives are practical, especially with fluorine and carbon, 2 hours and 20 minutes. Our scanners are such, we produce quantitative images. We're doing those great assays of radioactivity. And then, of course, what the topic of today is how we can apply tracer kinetics to take the beautiful radioactivity images and turn them into physiology. So in principle, with the right tracer, the right radiopharmaceutical, the right scanners, a reasonable human imaging protocol, we should be able to measure virtually anything. And this is, for those of us who've been doing PET for a long time, is what makes us excited about the opportunities that we have to open new doors and new windows into human physiology and pharmacology in normal brain, normal tissue throughout. I say brain here, but it really should be applying elsewhere. And really the trick that we have to battle is the fact that our images change with time. So here's an example of some brain slices with a serotonin transporter imager. And I'm showing you the early image from 0 to 10 minutes, where we're seeing a lot of blood flow information in the brain, higher binding in gray matter than white matter. And as time progresses, because of the particular tracer here that's binding to a selective serotonin reuptake inhibitor, we get more and more binding in the particular target. In other words, the flow is the initial part, and the particular binding, the particular target there matters. So we get time varying information, flow information early, target binding, this SSRI, reuptake inhibitor, but there's always that overlap. How can we best use these data? How can we extract that quantitative information from it? And that's where modeling comes in. We're going to take our dynamic data. We're going to have to have some kind of model, and a model is an algorithm or a set of equations, a set, or even a deep learning methods nowadays, usually with additional information, ideally the input function of the tracer, which I'll belabor terribly in this talk. And the goal is to separate things out so I get images, or at least numbers, separating out the blood flow parts from the particular binding or the particular target or metabolism that we're talking about. So when we embark on PET modeling, we're really trying to understand that relationship, those time-varying effects between the tissue data and what's going on underneath. We're really looking to account for the differences in the availability of our tracer, which certainly varies from subject to subject on different days. And then we're kind of putting on our engineering hat and say, what can we measure? How should we design our study? Then we move into validation, prove that we're measuring what we claim to measure, and verify that we're not bothered by other extraneous factors. Ideally, we can provide images of those parameters, those parametric images, not just radioactivity, but a binding measure or a flow measure, and then use that information to simplify. What can we do to make this a practical protocol for patients? This is very different. The kind of approaches we're going to take will be based on classical compartment modeling, but it's really different compared to what was classically done. Classically, you might have said, well, we can make measurements in the blood, maybe measurements in the urine or feces. And we develop models like this, where you look at the whole body and look at how everything interacts. And you have a lot of parameters to estimate, many compartments. And usually, the whole process gets pretty complicated. You end up making a lot of assumptions. And in PET, this gets simpler. Why? Well, we're directly measuring the tissue of interest. And at that point, if we have some idea what's going on in the blood, we don't really care about the rest of the body. Now, the other downside is we'd like to be able to make this measurement in very, very small regions, which means in general, we have higher noise. we tend to use simpler models. And when we talk about a compartment here, we're not talking about the liver or the kidney or the blood. We're really talking about subspaces like bound or free. And we use simple nomenclature, these rate constants, to describe those steps, being able to do that. Okay, so let's talk about the basic modeling methods that we use in terms of analysis. And since it's the first lecture, I'm really trying to not dive into them in details, just give you a flavor for those things. I'll talk about it in the world of neuroreceptors, where we are going to be talking about within the brain, where I have neurons that project into different areas. Often we're looking in the area around the synapse between the incoming axons and the receiving values, the dendrites. And if we zoom in further, we might be binding to a transporter, we might be binding to a receptor, we might be sensitive to synthesis, and we can produce images based on those particular update with the right pharmaceutical. We might write a model like this, where our tracer would enter the tissue. It might bind to the thing we're interested, the receptor. It might also bind non-specifically. In the blood, it will bind to proteins. And we always have to worry about metabolites. That is, the molecule, once it's injected, may be metabolized. And what happens to those? Are they in the blood? Usually. Are they in the tissue? Depends. So some words we'll use: the input function, that's the time course of the tracer in the blood. The tissue response would just be what's going on in the tissue, that time course that responds to that input driver. And because we cannot differentiate the different subcompartments, we're measuring the sum, which means sometimes it's hard to separate those compartments. We really need elegant data. And again, as mentioned, if we can measure the input, measure that blood availability to our target organs, we don't really have to care about the rest of the body. And that works very well in things like the brain and the heart, a little more complicated in something like the liver with 2 input functions. So what is it that we can measure with all of that? Well, if I took that model, we can then ascribe rates. We can ascribe numbers to each of those steps: the uptake of the ligand into the tissue, the binding and clearance from a receptor, perhaps even the non-specific binding in the case of a neuroreceptor model. What's the math? Well, this is for somebody who's been doing this a long time. You know, I learned pretty much all the math I needed in my sophomore year of college, learning about convolution integrals and solving ordinary differential equations. And these are linear because we're working at these very low tracer doses. We're not saturating any proteins or enzyme, which makes this a little bit easier to deal with. And then the job becomes, how do we interpret the values we get? Are they related to blood flow and blood volume? Are they related to a metabolic step? How do we take those apart? And often what we find is that maybe the parameters, the individual parameters, might be a little more variable. Maybe we can find something that's a little bit more reliable, a little bit more useful. And we've done that. I'll give those examples in a few different cases. So when we embark on this process of developing a model, we can certainly say which model, how many compartments, what configuration, what are the parameters. The harder part, what do they mean? Do they mean what we think they mean? We are very good at writing down these box drawings. To prove they really mean that is hard. Then to back off and say, what can we measure reliably based on the quality of the data, based on the duration of the study? And therefore, how do we optimize the data that we need to be able to do that? All right, so let's walk through some of the standard methods that people use. Here, we're going to be talking about 2 different classes of tracers, which is a similar way we talk about it: a reversible or irreversible. Reversible would mean if the tracer enters the tissue, it's going to be able to reach or at least approach equilibrium between all the compartments and what's going on in the blood. When we talk about irreversible, we're talking about something that's trapped, or, well, mostly trapped during the time of the scan. And this is really a function of time. If you go long enough, mostly everything will eventually clear from tissue. So it is a matter of time being able to do that. Now, for reversible tracers, we're interested in our delivery, our K1, how much we go from the plasma into the tissue. That tells us something about blood flow in some cases, depending on how well the tracer extracts. We're also very interested in the volume of distribution. We call it V sub T, also sometimes called the distribution volume. And it's a ratio. It would be, if we were at equilibrium, it would be like if we took out the tissue and incubated, what would be the ratio of the activity in the tissue to that in the blood? But it includes everything that's going on, whether we're specifically bound or free. We might look at a ratio of that between different regions. That's a handy way sometimes to simplify. We look at the volume of distribution in the region of interest we care about versus some reference region or organ. And sometimes, if we can be more specific, if we're binding to a target protein or an enzyme, we'd like to know its component, that binding potential, which we can get. We can relate back and forth with those distribution lines, these things there. And then we can tie these numbers that we can extract from the PET studies into the physiological parameters like receptor concentrations. Lots of ways to get at these numbers. That keeps the modelers gainfully employed. We can just do it the hard way. Collect the input function in the artery, measure the scan data over time, reconstruct the images, generate these time activity curves in some kind of calibrated radioactivity unit, do a fit to various models, and extract those parameters directly from the model. And we might be able to do that voxel by voxel. Here's that example I showed before where we have the dynamic images. Here, if we look at the parametric image, we've separated out the delivery, K1, from the binding volume of distribution. You notice we've paid a little price. These images look a little noisier, but we've separated that where no single time point here would be completely reflective of this physiological parameters that we're more interested in. If we can avoid the arterial blood, that's a great advantage. And the reference tissue model and simplified models are great ways to be able to do that, recognizing that the blood is the common input to all the tissue. There we can measure the binding potential, the relative or the distribution volume ratio between these two, and some relative delivery. We no longer get the absolute delivery values there. Or there's simpler models, graphical analysis, the methods of Logan et al., which basically find out we're not going to worry about the exact model and the exact configuration, but by a rearrangement of the data, we can produce data that's a straight line where that slope is that volume of distribution. That's a nice simplification, which allows us to do that for a wide variety of tracers, even when things are more complicated. Or perhaps we can infuse the tracer. We said this is the value at equilibrium. If we can actually get to equilibrium, which we can for some tracers, by infusing the tracer, we can immediately read out those volumes of distributions or binding potentials directly from the data. But this one doesn't always work. It really depends on the tracer and the duration of the study we're trying to do. Now, then we might have a pipeline for our brain pipeline. You might have an MRI and a template, which you'd register to the PET, that you could automatically generate time activity curves, plug them into the model to get these measurements like binding potential, extract the regional value. So you can have a very nice automated pipeline in many, many cases there, once you've characterized a tracer to be able to understand it. And one other aspect that's worth mentioning for the receptor studies is, well, we're sensitive to the available receptors there. So we can look at how they change in a disease, but we can also look at what's going on with either outside drugs or the neurotransmitters, because since we're working at tracer levels, if the receptors are occupied, we cannot measure them with a PET study. And so here's that cartoon. If you have the dopamine receptors, you have dopamine in white binding to their receptors. If we had our tracer, in this case, raclopride, if we increase dopamine here with amphetamine, there'll be less binding sites available. We get a reduction in that signal. And that's made PET very popular with the pharmaceutical companies when they wanted to be able to look at binding. Here's a study where we looked at a histamine receptor here at baseline. We give a drug that competes, and the same subject a few hours later shows much lower signal because there's just less available target. The same subject comes back next day. By now, the drug is clearing. We're partway back. And we can then build this beautiful calibration between the drug concentration and the occupancy, all being driven by the quantitative tools that we're using, so that the measures we have are not just radioactivity, but the physiological measure of occupancy, comparing post-drug to the pre-drug levels in each subject. Now, the alternative is to talk about irreversible tracers. And here I'm just really want to focus on the most commonly used tracer in PET, which is FDG, fluorodeoxyglucose. And for me, that always goes back to somebody old enough to have actually been able to meet and know Lou Sokoloff, who invented deoxyglucose. This was originally derived for autoradiography studies in rodents, and then was transcribed by converting tritium to fluorine to be able to look at that. With that really insight that if you labeled glucose, it would just go down a very complicated pathway. But by altering the chemistry of that, by changing the 6 position from the original hydroxyl group, that you can make the process similar by trapping it, or effectively trapping it, at least in most tissues there. And so here's what that looks like. Our model might say we take FDG in the plasma, go into the tissue. It will get bound into the tissue. For example, that could be tumor. And depending on the tissue, we may or may not have dephosphorylation. Here's what it looks like in normal brain. Here's the input curve, that is the FDG in the blood, the time activity curve, the initial arrival, followed by clearance. Here's the uptake into the brain here over 2 hours. We can measure the parameters of the model. Here's what they look like. But what we've learned over time is that the variable that's most relevant, that's most interesting, is this net uptake rate. And what that's reflecting is the original delivery into the tissue, K1, and then once it gets to the tissue, what fraction of it is bound based on that ratio, K3 over K2 plus K3. We can use that to calculate a metabolic rate of glucose. And it also means that we can be quantitative and get the most relevant variable, because honestly, getting K2 and K3 reliably out of these data can be challenging. And so this was formalized by Cliff Patlak, who basically said, okay, what we want to do is, can we directly measure that net uptake rate, not just the individual parameters, but with a simplified method to be able to go directly to measure that net uptake, which tells us about glucose metabolism. Here's the formula that he derived in his elegant paper in 83, and then the follow-on in 85. You look at the tissue to plasma ratio, you get this interesting variable that has the units of time. Cliff used to call it funny time, and that the slope of that is that Ki. And in fact, the Logan plot that I already used was motivated by Cliff's earlier work with this value. So you basically take the tissue to blood data as the y-axis, you take your blood measurements as the x-axis in this way, you have a simple linear relationship, and that slope is what you're going to care about. So it's something that simplifies that process. So if I have that data, here's an example with the same data, tissue to plasma ratio as a function of that funky time that Cliff used, and we can then measure that slope and get the Ki out of that. That's opened up a lot of things where we realize, yeah, there's more information we could be getting down here, but if we can simplify the process, we can get, in this case, the important parameter that we want, that net uptake rate. But you know what? We don't do that. It'd be nice if we did that routinely. A few places are doing that. It's actually, tools for that are being delivered into PET scanner code, but we use SUV. And we use SUV for everything. Certainly looking at that in the tumors. We use it for separating patients, for looking at changes over time, or looking at the hot lesion, all sorts of values for that. Is that a good idea? Didn't we learn anything from all those modeling studies? So what's SUV? Well, for those of you who hope not familiar with that, it says if I took my tracer and distributed it uniformly into my object or person, if it was uniform, I'd have a value of 1. If I have a hotspot beyond that, it would have a value which would be therefore independent of how much I injected or how large the subject would be. So are those related? Is the SUV that we use clinically all the time somehow related to this slope that we can get in the Patlak plot? It sure would be nice if that was the case. Well, let's do a little math here. For those of you early in the morning, I apologize. Here's that Patlak equation again, tissue over plasma. Here's that integral of the plasma over plasma times that slope, this value up here. And this VE, this intercept term, and that kind of reflects the non-metabolized binding of our target of FDG. So again, Ki is the slope, VE is that intercept. Let's make an assumption. Let's assume, so let's rearrange the equation. We'll multiply through by the CP. We get the simpler word. The tissue equals Ki times this area under the curve. That's the AUC of the blood plus the intercept. Let's start with an assumption that the intercept is small. And relative, for example, in high binding tumors, that's a pretty good assumption. It's not always an assumption. So it's one of those approximations we're making. So if we have that, at that point, the formula gets much simple. Let me rearrange. The Ki, the thing we like, is approximately equal to the tissue over the area under the curve of the plasma. But what do we measure? We measure SUV. Well, that's the same numerator, the tissue concentration. The denominator is different. We normalize it to the injected dose per body weight, body volume, perhaps lean body mass. So the SUV is in good agreement if these two things in red are agreed. So if the area under the curve of the blood is proportional to the dose per body volume, then the SUV is proportional to the Ki. And this has provided that quantitative physiology basis that said under certain assumptions, certain conditions, SUV can be very useful. And clearly it has been shown to be very useful, albeit a little more variable, probably more challenging when the intercept is more significant for lower uptake regions, and perhaps if there's anything that affects the clearance of the tracer. For FDG, that's pretty robust. When we apply that for other tracers, we have to evaluate all these ideas. So we know this in theory, we've learned that from the modeling, and the trick is how we apply that in practice. All right, so now let's back up a little bit and talk about how we develop those tracers in the first place. And I will talk about my favorite tracer these days, which is carbon-11 UCBJ, our synaptic marker. You know, there's a lot of steps one goes through in developing that. You have to figure out what target. Are you binding to a protein? Are you binding to a receptor? What is it that you're after? We'll probably do some in vitro evaluations to tune which chemical matter we're looking at, develop the radiochemistry, get that approved for human use, do the dynamic scans with arterial blood samples, do the regional analysis and the modeling. Look at how reliable it is with retest studies, if there's a drug that you can use to block it, and then to absolutely validate that, where you're comparing the in vivo data, ex vivo or in vitro data. The target we're using for our synaptic is a protein on the synaptic vesicles, which are in the presynaptic terminals, called SV2A. It's been, other of these proteins have been used for years as measures of synaptic density, synaptophysin in particular. Here's what the initial images look like for this tracer. This binds very heavily in all gray matter regions. Here's the time activity curves for a test and retest, and we certainly find very high uptake. SUVs of 10 mean we have very good quality data with nice clearance in the reasonable amount of scan time. We measure what's going on in the arterial blood. So we look at not only the total activities in the blood, but we look at the metabolites. At early times, we have one molecule, the parent tracer. But over time, the tracer is metabolized. And we find out what fraction of the tracer can be attributed to our parent compound. This adds some work to do. We have to decide, what about these metabolites? Do they cross into the tissue? For the brain can be very restrictive on these things. We don't have that luxury outside the blood-brain barrier. We do the modeling. We compared a couple models. This one was fortunate. That's a very simple model with an uptake rate and a clearance rate. Describe the data very well. Good agreement between that one tissue model and the more complicated model. And that means we get two variables of reading out with this tool. We get our volume of distribution. That's that equilibrium ratio. That's the thing we care about. That's related to the amount of that target protein related to the amount of synapses. We get like values of 20. It means 20 to 1 against the blood in the gray matter, much lower in the white matter. But we also get a K1. We get 2 for the price of 1. K1's telling us something about the delivery, about the blood flow. And it's not equal to blood flow because this tracer doesn't easily cross into the tissue as well as, for example, say water, but we are getting something that's sensitive to that, as I'll show shortly. And then we can do that analysis. So what we've done now is took those time activity curves for every pixel, fit it to that model, that one tissue model, and now I'm showing you that volume of distribution on a voxel by voxel level. It's very exciting if you have that opportunity because that means we're in the position to be able to look at very, very small regions with very good accuracy. Alternatively, it means we could be lowering the injected dose. We can do our scan time. So we, for example, here we collected 2 hours of data, and we can compare the results simply by chopping off data in the time activity curve, comparing that to 2 hours of data. And we find 60 minutes, the differences are so minor in our different regions of interest that a 60-minute scan is more than enough. Looks like we're starting to get some variability down to 30 minutes. So certainly the 60-minute scan is sufficient. And then because this target, SV2A, is also the target of a drug called levetiracetam, also known as Keppra, an anticonvulsant, which has been given legally to humans, we can demonstrate that in vivo that we can displace it. So all parts of that job to say, how do we measure this target? Are we in fact sensitive to that? And then finally, we do the validation. This we'll do in large animals and baboons. We've done the in vivo scans, and we can then do that validation by extracting the tissue by Western blots, look at ligand binding assays, and confocal microscopy. We were able to show good agreement between the in vitro data and the in vivo data. This is a fortunate system where it worked very well to be able to do that. Ideally, this is something we would do with every tracer to really put us on firm ground that we're on the right track. So now let's talk about how we use these ideas, how we've gotten some models, we've gotten a tracer, we've gotten some models. What do they do for us? How do they help us to extract information from the data? How do they help us simplify? We work with that. So let me take this first one, and I'm going to follow on with our SV2A tracer, UCBJ, to separate out that blood flow and binding to that SV2A target that we can do in that mode, okay? And this was really relevant to the physiological neurological question. We're binding to, this is the presynaptic terminal, we're binding to a protein in these vesicles. Well, the job of the vesicle is to bind to the protein, release its neurotransmitter. I mean, that's how you're thinking about anything that we're thinking about, is that when those vesicles are out here, does that affect what we're doing? Are we sensitive to neuronal activation? Or in fact, is our measure very, very simply a function of how much SV2A there is in each region of the brain? So what we decided here is let's activate the brain and being able to do that. Now, activation, of course, has long been shown to change things in blood flow. And here where we can measure both things, the blood flow related information K1 and the binding to SV2A and VT, we could separate those out. So we did a classic activation. This is our alternating checkerboard. It goes at 8 hertz. It's a great way of activating the back of your brain, the primary visual cortex. And so paired scans are done, one with baseline and one with this back and forth visual activation. And we also did this with fMRI, just kind of a double validation that our activation was working well. So here are what those results would be. Here I'm showing you the images that we got from there. I'll focus on, here's our volume of distribution image that's showing the binding to SV2A. Here's the K1 images showing the uptake of the tracer related to blood flow. And what you see is that really focal change back here in the visual cortex. That's the spot we fully expected to see. It's also called V1, primary visual cortex, we get this very robust change in all of these subjects, about 35% increase, consistent with that your brain was very, very busy processing that alternating checkerboard. Other regions of the brain, cortical regions, white matter, no change. In their VT, we saw no change in there, which suggests that by doing the modeling correctly, we can separate out the blood flow effects from the binding effects. And this suggests, at least in this case, that we were not sensitive to the activation. That can also be true with the binding potential measure. So it's this data, limited though it may be with one activation, suggests that by doing the modeling, we can get a functional assay, and we can also get kind of a more static assay, something that's more like what we could do if we did a Western blot of tissue in there. And we could validate that against MRI as well, although I don't know which is validating who. If we look at how much K1 changes compared to the bold signal changes, we actually got a good agreement across our subjects. You know, the PET signal is a big signal, 35% increase. The bold signal subtle, but the signal and noise of these two measures are pretty comparable there. So again, just to emphasize, we get this 2 for the price of 1 by doing the modeling, the binding measure, the VT or binding potential, the flow measure for this tracer. And that showed to be very valuable even in our Alzheimer's work. And I won't belabor that study, just to point out, when we looked at the binding measure there in that original paper, we saw a nice change in the memory area of the brain and the hippocampus. When we looked at the functional measure, not only did we see the hippocampus, but we also see things in regions that have been shown for a long time with FDG that are showing the functional measure. So a static measure of the protein, a more functional measure. The modeling lets us take those apart with one study, in this case, a 1-hour study. Now, how can we simplify? Understanding that, can we do things to make life a little simpler, more practical for some of our patients? So what are we thinking about here? We've got a model. We think we understand how it works, how it's related to the physiological action. We've done all that work we've talked about there. Maybe we can skip that blood samples if we can find a reference region to normalize, because those blood samples, it gets pretty invasive. And honestly, not that many sites are ready to not only take blood samples, process them, and also handle those metabolites. And then maybe we want to look for, instead of that dynamic scan, even those 1-hour scans don't fit into a lot of practical clinical assays, what's a good time period that might give us a good measure, where a simple measure that we can get from our data might correlate with maybe the modeling measure, the distribution volume ratio between our target regions and perhaps a reference region if we can obtain it. So that's our goal. We're going to do something. We're getting an SUV, but an SUV ratio, which is just the ratio of the SUV values in the target tissue to that in the reference ratio, that SUV ratio there, to be able to concentration ratio there. And then we can apply that. And boy, that makes things much simpler when you're thinking about clinical trials. And nearly every successful tracer has found a way to do this. But let's talk about that one. So here's an example with tau tracers, MK6240, published a couple of years ago, where people found an SUV ratios and a distribution volume ratios using different kinetic methods, but basically showing pretty good agreements if you pick the right sweet spot in time. And that's the trick. Each tracer, slightly different sweet spot in time. For our tracer UCBJ, same thing, and we definitely have time dependence. If we did 30 to 60, we're a little low. We did 90 to 120, we're a little high, and the Goldilocks time for UCBJ in terms of having a good agreement with the SUV ratio and our binding measures is from 60 to 90 minutes. And again, each one of those going on. So why is this? Why is this time dependent in this way? So let's think about this a little bit. So let's imagine we're going to do a perfect theory. We're going to do a little thought experiment here. Imagine I have two regions. I have a reference region. It has a volume and distribution of 10, which means at equilibrium, it would be 10 to 1 over the blood. Here's my target region. There's more of my target protein there. I'd say it would be about 20 to 1. So I'm looking for a value of about 2 if I wanted to calculate a distribution volume ratio. All right, so now I'm going to do a simulation. This is a really simple simulation. I'm just going to have no blood clearance. The blood is going to be perfectly flat right from the beginning, and we're going to see what's happened. Well, my reference region comes up and would reach its equilibrium value. My target region, well, it takes a little longer. Usually it takes longer to fill the regions that have more binding. And so here's my apparent, if I just took the ratio over time, by the time I get out to that period, 70 to 90, that's the perfect sweet spot. This is the case where nothing is clearing from the blood. But now let's make it a little more interesting, a little more realistic. Let's let the blood clear. Because when we inject it, you saw in those blood curves, the blood does clear. What happens now? Well, now I find I overshoot the reference region and I overshoot the target region even more. If I calculate the distribution volume ratio, just the ratio of these dots here, not only do I exceed the equilibrium level, this is the real number because that's what's going on with those proteins in that tissue, but it keeps going over and over. So instead of scanning at 70 to 90, maybe I just want to go a little earlier. And that's what our field has done. They've empirically looked for that time in a standard patient population where we can get the right answer. And of course, this is nicer there because not only does that time, this actually time fits better into clinical practice because often you're just scanning patients with a given tracer around the same way you're doing things with FDG. And in a study that Ansel Hilmer did a couple of years ago, the magnitude of that error, this ratio of the apparent volume distribution, the magnitude varies. It's a function of the plasma clearance rate, and it's a measure of what the volumes of distribution would be looking like. So what's going on? Well, if we give a bolus injection, we're not at equilibrium. And in fact, if we calculate that tissue to plasma, we'll eventually get to some constant, but it won't be the right answer because we're not at true equilibrium. The blood is clearing. And as it turns out, well, the bigger the plasma clearance, the bigger the effect we have. And if we have a lot of target protein, the bias gets bigger. We just saw that in that simple example that I showed. When we calculate the ratio, that's kind of the ratio between those two. And, that's overestimating too. If we got all the way out to that period, transient equilibrium where things get flat. So we can help by scanning earlier. Is that a problem? In other words, can two wrongs make a right? No, if we waited until we got to that constant level, we would overestimate, and that would be a function of the clearance rate. If we scan earlier, we can get the right answer. So we're overestimating if we waited to the equilibrium because we're not really at equilibrium. By scanning earlier, we underestimate there. Is that okay? What could possibly go wrong with that idea? And the trick is, will we always get the timing right? Is the cases when we evaluate that affecting always the case for the next population we use? Is it going to be a function of age, a function of sex? Will drug treatment affect it? And I'll give a little plug for a grad student, Praveen Hanhar, who published in the SNM earlier this year about a correction formula to deal with that. But even that needs to be validated in larger populations as well. All right, how about outside the brain? I'm doing a lot of brain work, doing some outside the brain. I've really come to appreciate the brain. The brain has a lot of nice features. Doesn't grow, doesn't shrink, doesn't breathe, doesn't beat. When it moves, it tends to be rigid, tends to have good blood flow. That's good. And it has that blood-brain barrier. And that means when our tracers metabolize, often the metabolic products are watery, hydrophilic, and they don't cross the blood-brain barrier well. So in many, many cases, the tracer in the tissue is in the brain is just the parent compound. That's a great luxury there. And that's hard when we go outside the body. We have a lot of things. We have to understand that input function about how do we measure the metabolites? What happens? Do those metabolites go into the tissue? And if we want to use a reference region, well, if I'm looking in a liver or a tumor, what would be there? And of course, motion is really challenging in the body, much more so than in the brain. Now, one thing that's great when you're scanning in the body is you've got access to blood. And when you have something like the Explorer, you always have access to blood. You have to pick where you're going to get your images, where you're going to be able to do that, what time to be able to do that. And if you're not going to collect things dynamically, you might need some kind of population model where you might be able to scale the image-derived input data, being able to do that. And this has come a long, long way, especially when you have body data with access to the aorta, which is very much bigger than the current resolution of a PET system there. But the metabolites can be tricky. Here's an example where we've looked at that for a marker we were hoping to use for beta cell mass in the pancreas. And in the blood, here's the metabolite profile. Lots of metabolites in the tissue here at 90 minutes. This is from baboon data. And then when we took out the pancreas of that baboon, we were at least finding 90% of the signal was the parent. That was fortunate. That's not always going to be the case in each different tracer. How well will you do? And it also affects what reference region we could use, because in fact, in the reference region with much lower activity, you do have noticeable amounts of metabolites. How do we correct for that? Do we need to? And in picking those things, it can be really challenging to pick a good reference region. Here's what we really like. It'd be perfect, where I have a region which has some level of non-specific, non-displaceable binding. That's exactly the same as my target organ. And then the specific binding to the protein just sits on top of it. It's a beautiful world if this happens. It's hard to do it, harder to validate it. Maybe we can do it in large animal. Very challenging to do that in humans, unless we have a good blocking drug to be able to do that. So now some examples I'd really like to push through in terms of when the trade-off between not modeling and modeling. And I really call your attention to this great paper by Adrian Lemertzman, now it's about 5 years old, about really looking at the comparison. Here's one example where we looked at just retest for amyloid. If they just use an SUVR, that value on average, there was no change in this population, but it was much more variable than when they used the modeling measure here, looking at the DVR. And again, you see the little overestimate that SUVR was doing for DVR. Or here's an example in the heart. very commonly in myocardial work there, you're looking for a regional defect. And you might have studies where you have a baseline and you have a stress there. And honestly, if you just look at that parametric, without looking at quantitatively, all you'd be looking for is, are things non-uniform? Are you seeing a regional change in that way? But in this patient, for example, if you looked at that, absolutely, and again, the scales are very different here, there was no focal defect, but the relative flow was not there. Because if you only looked at relative flow, you wouldn't miss the fact that they didn't get the high flow increase that you needed. So the absolute measures were very important. And cardiac's a really important area where that is something that we can do, certainly with the right kind of tracer. But I want to focus on thinking about the input function, the availability. We're kind of running under the assumption that when we inject this tracer, that everybody processes it the same. The only difference is how much we inject and maybe what's your lean body mass. But boy, when we give a drug, that changes. Here's the most extreme example that I'm aware of. Here's that serotonin tracer again, looking in the brain. If we did a blocking study with citalopram, that's an SSRI, that blocks the sites. Two big things happen. And the trick is with this one is that there is a lot of SSRI in the lungs. And if you go in the lung, so normally the lung, which gets the first cut of any IV injection, is going to take out a lot. But if we block all that site, there'll actually be more in the blood because it gets through the lung. And because the lung is not there to resupply it, it clears much faster. So it looks like you've got great blockade in the brain, which you do, but it's an area where you might want to be thinking about how to interpret that. So when we do these studies, what we'd like to do is get this nice dose response curve, being able to do that. And the trick is that when we block the drug, as we just saw with AFM, we increase that bioavailability. The non-specific uptake is higher, and we have to balance those two effects. So let me give a couple examples, and I think I may skip through in the interest of time. So here's a study from Ansel Hummer where we were looking at neuroinflammation here in a monkey brain. We activated this with LPS, and we got a nice increase in our binding. This is a PBR28, which returned back to normal in 14 days. Well, we gave a blocker, which depleted the microglia in the brain. We get a reduction and increase. Big decrease when we block, big increase when we activate. Now, what if we didn't use the modeling? What if we just looked at the SUV values there? Well, we still see the pattern, but we get the magnitude wrong. And why is that? Well, by giving those either the blocking drug or the activated drug, it changed the input function. And that hurts to reduce the magnitude of signal. If I block this everywhere, what it will mean is that there's actually more blood available for this, just like blocking a brain study. And so the non-specific uptake here is higher. We underestimate the effect there. Or here's an example with an enzyme inhibitor study. Here's just the SUV images here. This is a monkey. We did a baseline scan. We gave a blocker. We expected it to show less signal, but wow, it went up. Now we went to a higher dose, then it went down. Really scratching your head on this one. And the trick here is what's going on in the blood. By giving a blocker, we block this enzyme all throughout the body, and what we got is a huge increase in the blood. And so what that meant, going back to that slide, just the availability, because there was more tracer available, it went up. When we went to the higher dose, by then we had blocked it mostly, not much of a change. If I correct for that, here what we're showing are Patlak images. We've done the Patlak analysis. This is an irreversible tracer. We get a small reduction in low dose and a high reduction in high dose. And so that's a good example of how those things would work. All right, let me just take the last couple of minutes and talk about where we are going. And like I said, I cannot resist the opportunity to talk about our next generation brain imager. We talked about this in the beginning, what we need, the right pharmaceutical, the right imaging technology, a practical protocol, and we could measure anything. Well, what are the limits in this? And with the next generation of both instrumentation and boy, nowadays, algorithms, machine learning in particular, what can we do to expand that scope? Well, there's a lot of things we got to think about. When we look about there, how much of the target do we have? I'm talking about the brain. If we have small targets, thinking about maybe alpha-synuclein and Parkinson's, it's a hard one. How much background of that in our tracers? How big is the region we're looking at? Are we looking at a tiny region or a big region in the brain? And how efficiently did the tracers go in? All these things affect our signal. And then we also got to look at the kinetics. How long does it take for our tracers to approach that equilibrium? We can get that binding. How can the patients tolerate this? I mean, if we're going to try to study adolescents, for example, if we're looking at changes in disease, is it a 50% change or a 5% change? If we're trying to follow things over time, is it a 10% change or a 1% change? And we'd like those protocols to be simple, but not just need to be in the most advanced research centers, but to be practical in hospitals. That's how our tools get out there to have the biggest bang for the buck. So here's some examples of things we're doing now. I mentioned in that synaptic marker in Parkinson's disease, the target that really hurts is an area in the midbrain called the substantia nigra, or the source of your dopamine neurons that we published. And we could see that group difference, but what we really like to do is to follow these patients over time or see what drug treatments are. These are small signals in very small regions. Or maybe we're trying to look at dynamics. Here, where we're looking at a tracer that binds, where we can give an activation to get the change. So if we gave some kind of activation, some kind of signal, can we differentiate that? Do we feel like there is an effect or there isn't an effect? And that really depends on how good our signal is to use even more advanced modeling to be able to look at that. This is the LPNT PET that Evan Morris promulgated. So this is the scanner we're developing right now that we're expecting delivery late this year, early next year. It's going to have a big axial field of view around the brain to be able to do that, really inspired by the whole body explorer work. We call it the Neuroexplorer, the NX. Small detectors, so we can be able to measure fine resolution, very good time of flight, and very good depth of interaction that we need to do. And we're separating out the PET part from the CT so we can really get in there for those blood sampling studies that we need to be able to do. And so what might these tools help us with? Well, better statistics. We'll surely get lower noise. That's a good thing. We might improve the detection of those dynamics like we just saw. Maybe we could scan longer. I mean, some of our tracers, here's a kappa agonist tracer that we are really barely getting the equilibrium even out 150 minutes. Well, as been shown on the Explorer, higher sensitivity buys you time for carbon level. And then the modeling can even rank up to the next level. Can we start really trusting those microparameters without having to constrain them? Can we do multi-tracer PET? It's been an idea around for there. Can we do that more accurately by handling those two tracers, be able to get something in a more patient-friendly way? And sometimes our tracers are a little dirty, and maybe we can understand that they're binding to two targets. Or for tracers where the metabolites go in the tissue, can we take that all into? And of course, in a brain scanner, we'd love to measure that input function. Here's what the carotids look like on our HRT. And we'd love to be able to understand that. We still have a lot of work to do because that measures the whole blood activity. We've got to separate out for plasma and separate out the metabolites. So still a lot of work to do with that. So a couple closing thoughts. So first, modeling methods, they really permit us to take it to the next level, not just get measurements of radioactivity, but really take this up to look at the physiology and pathophysiology in vivo with great accuracy. That's the good news. And with that good basic science, I mean, the whole is, how can we turn that into clinically relevant imaging methods? How to make that simple. But on the other hand, the modeling studies are often way more complicated, doing arterial lines, and they're costly. And so you want to find a way of getting that where that translates your understanding into something a little bit simpler. And honestly, you don't always need that great accuracy, especially when you're looking at within subject changes due to drugs or just the normal treatment effects that might be there. So the real trick is to use what we understand from the models to develop that simpler, more patient-friendly assay that's going to be practical in there. And just keep it in mind that they come with a lot of assumptions, and we're really good at forgetting about them, like what we talked about with the SUV versus the FDG-KI, to remember, are they relevant there? And finally, this next generation of instrumentation algorithms is really going to let us go back to the future again, go back to the things we started looking at a long time ago. Let's revisit them and see what additional information that we can get. And so finally, just the acknowledgement of the team at Yale, the Neuroexplorer team, and the pictures on the front, and all of our funding sources. And I thank you for your attention.